Thank you. Thank you, Peter, for that introduction. And I'd just like to thank the organisers for inviting me to speak this afternoon and to say it's an absolute pleasure to be here in Portland. Right, so the title of my talk, as you can see, is Whole Genome Sequencing Has Transformed the Detection and Investigation of Outbreaks. And I'm going to spend a little bit of time at the beginning going over what we've been doing in the reference lab I'm head of. We've chosen to go the other way round in terms of applying whole genome sequencing to pathogens to CDC, and we started with Salmonella. So I'm going to give you, show you what we've done, and then I'm going to give you some examples of using whole genome sequencing for some outbreak investigations. Okay, so I'm head of the gastrointestinal bacteria reference unit. It's a national reference laboratory, and we get sent isolates, and you can see the range of pathogens here, and Peter also mentioned them. So ranging from Salmonella, E. coli, gram-positive foodborne pathogens, and also Campylobacter and Helicobacter. So we are referred isolates from food and water laboratories, so from food samples, and also from clinical isolates. And probably most of the isolates we get are from clinical cases, but we do get both. And the aim of our work is we are identifying those isolates that are sent to us and characterizing them. So we're using typing methods. And this enables the frontline labs to get a confirmation of the organism they've isolated, and it can confirm the diagnosis in a patient. And we also do it's also done for national surveillance, so we're looking at the monitoring of the infections that we get, and we also do typing for outbreak detection and investigation. Okay, so PHE's had a huge investment in whole genome sequencing. It started with our business plan in 2011, where we thought it was the right time to invest in this technology, even though it's very fast-moving and changing all the time. Currently, we have 2 MiSeqs and 2 HiSeqs, and we also have this machine called a NextSeq, which is in between both of those. And we have the capacity to do more than 3,000 genomes per week. We also have a machine called the MinION, which we have on loan to us, which is one of the machines has been talked about already, that you can just actually plug into a laptop and get a full genome sequencing in about an hour and a half. So we're just sort of playing around with that at the moment. Okay, so I said I was going to just go over what we've been doing with Salmonella. This is just to say what we previously used to do, how we used to work with our isolates. So we receive about 10,000 Salmonella isolates per year, and as I'm sure you all know, there are 2 species of Salmonella, Bongrai and Enterica, but there are 6 subspecies of the Enterica. And then Salmonella are characterized by serotype, and there are over 2,600 different serotypes of Salmonella, so it's quite complex. Our isolates that come to us, we speciate them and subspeciate them. We'd use an initial PCR assay that identifies the subspecies 1 or the Enterica. Some of the other subspecies need to be confirmed in their identity, and we do further biochemistry using a phenotypic microarray-based system, the Omnilog. Then the isolates are serotyped using traditional agglutination reactions, according to Kaufman and White, using based on the ONH antigens. Then in our reference lab, we still do phage typing, because we find that's quite actually a quick method of typing. And then If we see something that's suggesting an increase in a serotype and phage type, we will then go and use our molecular-based methods, so MLVA and sometimes pulse field gel. But we don't do molecular typing on all isolates because we actually just have too many isolates coming in. And we also do antimicrobial resistance testing. So the question was, can all this be replaced by whole genome sequencing? And why did we want to change? Well, we wanted to change because the traditional methods are quite lengthy and we don't, unable to do all the molecular methods on all isolates. We know that the existing typing methods don't really give us a true biological classification. And they can't tell us, if you've already heard earlier this afternoon, that they can tell us that strains are different from one another, but they don't tell us anything about the real genetic relationship or how different they are. We also had problems in the reference lab that some of the isolates referred to us were sent to us as hazard group 2 organisms, where in fact they were hazard group 3. So they were sent to us, and while we were processing them at category 2, we found out they were Salmonella typhi or paratyphi. And that means laboratory staff have been put at risk of exposure. So we thought whole genome sequencing might be able to help with that. We also thought in terms of quality, using a single method would help in standardizing the procedures, help us to get increase reproducibility and enable methods to be compared with different laboratories. And also quite a lot of pilot projects we did indicated that whole genome sequencing would really help us in identifying outbreaks and clusters of cases. It will provide better evidence for traceback and identifying the vehicle and the original source of contamination. And it would enable us, and for all those things, it would help us understand outbreaks and ultimately lead to better prevention and control interventions. So the idea is that whole genome sequencing can provide a single one-step method for identification and typing, but not just that, it provides a whole wealth of other information. And we actually don't use all that information in there. There's a huge amount of information that is there to be used by other scientists, biologists and bacterial physiologists who can answer sort of basic biological questions. So it's a kind of win-win situation. So this is our sort of Salmonella workflow. We have DNA isolates come in, DNA is extracted, they go over for sequencing, the sequencing files come back to us, and we perform bioinformatic analysis. And that it can be separated into two things, although it's sort of done at the same time. These pipelines run at the same time, and we can get identification and characterization and typing, and to replace the phenotypic methods that are in black on this slide. So I'm just quickly going to go over how we can do this. It's previously been shown by Achtman et al. That multilocus sequence typing of Salmonella, so that's your 7 housekeeping genes, correlates very well with Salmonella serotyping. And so it was quite easy for us to develop a pipeline that pulled the sequence data from whole genome sequencing, and we are able to assign the sequence type, so infer the sequence type. And as you can see in this slide, in our validation phase from April last year to this year, we did nearly 8,000 isolates, and we found that we got a 97.2% concordance with traditional serotyping results from our sequencing. Now, the small percentage that don't correlate are due either because sometimes the sequencing quality isn't isn't sufficiently high, but as you can see, that's quite a very small percentage. Sometimes our isolates come in, we receive them on slopes and they're mixed, so it might be with another, not even with a Salmonella, so you don't get good data from that. But probably the largest reasons for not getting a sequence is because in the database that exists where you can, that is of sequence types and serotypes, the serotypes haven't got a sequence type yet. So we're actually describing a lot of novel serotypes, and we're adding them to this database. Okay, so that's having told you about how we assign the actual sort of identification of Salmonella. We're now going to talk to you a bit about the outbreaks. So we use, in Public Health England, we use an exceedance above what you would expect to see as background to trigger an investigation into an outbreak. And that will be based on serotype and phage type. And it's only when we see that level increase that we would then look to perform a molecular method. And if it's a Salmonella enteritidis or typhimurium, we would use MLVA. And if it was another, depending on the serotype, whether we would need to do pulsed field gel electrophoresis. However, although that gives us greater discrimination, we're not doing it on every isolate. And the more common a serotype is, such as enteritidis or typhimurium, which make up the majority of the infections in the UK, the harder it is to spot an outbreak. So we decided to look at using SNP analysis, which you heard described by David, to look at the detection of clusters and outbreaks. So what we decided to do is that for each of our the different serotypes, and there are 90% of the infections in the UK are caused by about 10 common serotypes. And as you can see in this slide, Salmonella enteritidis and typhimurium do cause the vast majority of infections. So for each of the serotypes that we get identified by MLST, we've generated a SNP database. And this produces a, I'm just showing you very quickly on the right-hand side there some just the SNPs, but you had that explained to you before. And the analysis, as I said, it's run very quickly. So as soon as we get our MLST result sequence type, it will assign the serotype. It goes into a SNP database, and it does a phylogenetic analysis. And we've generated what we've called a SNP address, and that tells us how similar any isolate is to the nearest one in the database. And we can use it for describing outbreaks or clusters using this SNP address. Okay, so again, just going over back to the whole workflow, the Salmonella workflow, the isolates are received by the laboratory. We carry out automated DNA extraction. We send them over to our sequencing unit, and they send us back the data, the FAST-Qs. We have a bioinformatics pipeline. We use the KEMA ID, which that really tells us, have you got what you think you have got? So we're sent Salmonella isolates, but sometimes they're mixed, and that will be picked up by this KEMA ID, what actually is there. Then we have a pipeline that calls the MLST, the sequence type, and assigns the serotype. And then the strain goes into our automated SNP analysis, the database, depending on its serotype, to look for the different clusters. And that now triggers the investigation. So first outbreak I want to talk to you about is one that occurred last year. It was an international outbreak of Salmonella enteritidis phage type 14b. And in England, we noticed there was seemed to be a point source outbreak in a ward on a hospital in Birmingham, and it was a single MLVA profile. But while this was going on, we also noticed around England there were other smaller point source outbreaks going on that were related to restaurants, different eating establishments. And we also had some isolates. We had some isolates from the hospital ward that came from, obviously there were patients, but there was an isolate from a food trolley and from a cloth. We also had some isolates from some of the restaurants from food and from chicken and pork from one restaurant in a cloth, and from another restaurant it was associated with egg noodles in the restaurant. So this is showing you the whole genome sequence analysis of this outbreak in the UK isolates, and down here is the hospital outbreak, and they're very, very similar. In fact, across all of these, there's only about 0 to 3 SNPs difference in those outbreaks. So they were showing, although they were point source outbreaks, they were all related, very similarly related. And that's actually just putting on isolates from 2012, which, because of their similarity, indicates that something probably had been going on, but we hadn't been aware of it. So it probably started back in like 2012. So, as I said, this was an international outbreak, and France was the first country to put up a European alert system, epidemiological alert, that they'd seen 6 outbreaks of Enteritidis PT14b, and they had an egg isolate with a German egg mark. And this had the same MLVA profile that we had seen, or it had a single locus variant, which indicates that both the single locus variant they are linked with the original profile. There were also cases in Austria, Luxembourg, and Germany. And so the traceback investigations led to sampling of a single egg production premises in Germany. Well, actually, it wasn't a single egg production. It was a single company in Germany that owned 3 different sites in Germany. And these 3 sites had the same were the same feeder flocks. They were the same laying flocks from the same one supplier. And in the UK, we were also able to trace back that all the hospital outbreak and the other outbreaks were the German company supplied eggs to the catering establishments and to the hospital. So it was the same German egg supplier in all cases. So their sampling was done. The eggs in France came from one of the sites. The eggs in Germany came from sites 1 and 2, and eggs in England came from site 3. So because there was a big international outbreak investigation and outbreak control team, all the isolates from other countries were sent to PHE, England, to our lab for sequencing. And these are the results, the sequencing results that came back. And here, what you're seeing on this side is all our Salmonella enteritidis isolates. This is the phylogenetic tree of all the isolates. And we're just, on the right-hand side, we're honing in on this little red star here. So we're kind of zoomed in here. And as you can see, you've got all the UK, most of the UK isolates have clustered here in one cluster. Then you have another cluster here, which is red. These are German cases and German egg isolates. And at the top, in the blue ones are the French isolates, and also French egg isolates and French clinical cases. So, and all of these are related, and they go back. So across this whole lot here, you've got about 23 SNPs. And they all trace back to a common ancestor, which was about in 2012. So this is providing very, very good microbiological evidence for that for the traceback that we did, that it goes back to this German company with 3 different sites. So just also to show you at a local level, this is the traceback that occurred in the UK. This here represents the German producer, and it's quite a complex supply chain, so this is distributing to the different restaurants involved, to the hospital ward via various intermediate distributors. And around here, these are the number of cases or the number of isolates here. And what's quite nice is you've got this quite complicated traceback, but your phylogenetic tree is showing that they're all, this one is here, this one is here, this here, is showing that they're all phylogenetic, they're all related. So it's giving you, it's very clear link between your isolates from humans, from eggs, and from environmental samples. So what you're getting with the whole genome sequence data is that you're getting very good evidence that isolates are related to one another, but it's also putting them in a phylogenetic context, which we haven't had previously. Then Okay, I seem to have missed one of my slides. The next incident, outbreak, I wanted to talk to you about, and I seem to have just missed one of my slides here, was an outbreak that occurred we identified this year, that actually started, we were alerted by Scotland, who'd seen an increase in Salmonella enteritidis phage type 21 in February and March, and they had 5 cases. And they had one single MLVA pattern, and they contacted England and said, had we seen any isolates of this type? Well, we ran our SNP database on enteritidis, and we did find that we did have a cluster of cases from August of last year, gradually increasing with the highest numbers in November. And this actually also Salmonella enteritidis. It wasn't just phage type 21, it had related phage types in there as well, which we wouldn't have picked up under our usual exceedance system. And these isolates, as you can see here, all clustered together, and they're all within 10 SNPs there of that outbreak. So there were about 55 cases, and it went over quite a long time, and it was not triggered by our usual exceedance level because the number of cases was actually quite low, but it went over a long time. So this is just an illustration of something that we may not have seen previously. The cases were interviewed, but because it was over a long time, we didn't have good epidemiological data from these cases because they weren't interviewed immediately. We managed to interview 10 of them, and 90% of them had eaten chicken nuggets, so that's bits of chicken that have a sort of goldeny breadcrumb thing that's quite popular with children, and these cases were predominantly in children. And 10%, that was compared to 10% into a comparative group. And so this sort of provides evidence for us to investigate the potential contamination of chicken nugget products or potentially a single supplier to the various supermarkets. These investigations, the food investigations in England are done by the Food Standards Agency, and they did investigate this, but they weren't able to find any evidence of a single supplier or contamination. We feel that we need to improve the investigation of such outbreaks because they previously would have been under the radar. But we're seeing more of these, and we need to find a better way of investigating them. The final outbreak I want to go over is actually an E. coli 0157 outbreak that occurred in 2013. We noticed there was quite a sharp increase in this 0157 phage type 2, Shiga toxin subtype 2A. And we do, for 0157, we do have very good enhanced surveillance questionnaires. And most of the cases were women, and there were about 19 cases in all, and it became quite clear from the enhanced questionnaires that there was an association with eating pre-packed salads. And when we re-interviewed with a trawling questionnaire, it became clear that they were associated with watercress. Now, at this stage, we had 2 MILVA profiles. Some of the loci were similar, but some were different. And so we were quite unsure about was this the same outbreak, because there was watercress in both of these sort of MILVA associated with these 2 types. And were they the same strain or were they different? One of the slightly different MILVA profile, those cases had not purchased watercress from a major supermarket supplier, but they'd eaten watercress at a pub that was supplied with watercress by the same supplier who supplied the supermarket. So there was quite a big, because this was a major supermarket who'd supplied the watercress, there was a withdrawal of the product, and there was quite a lot of publicity about it. We did some whole genome sequencing of all the isolates, and what we found was there actually were 2 outbreaks going on here at the same time, both of them associated with watercress and associated with the same supplying company. Now, for the first outbreak, this is very clear from here that these are over 300 SNPs difference from one another. These 2 different isolates, clusters here. So it's very clear there are 2 different outbreaks. The first outbreak we had, or the outbreak 1, we looked at the isolates that we've got a big database of 0157, both from previous clinical cases and also from cattle in the UK. We couldn't find a match at all. Now, we contacted, our bioinformaticians know that the person at University of Texas had a lot of 0157 isolates from cattle here in North America. And it doesn't match with any of those, but it is more closer to those isolates than it is to the UK isolates, which sort of suggests that this could be an imported isolate. It's come from outside. And in fact, when further investigations led to us finding out that the seeds for the watercress came from North America and Europe, and that the peat for the watercress came from mainland Europe. Now, the second outbreak did cluster with our domestic E. coli 0157 strains. So it did look as though it came from within England. And then we started a huge investigation. There were 10 farms that supplied this watercress processor. And we sampled water and watercress from each of those 10 farms. And 4 of them had high E. coli counts, so we went in and did further sampling. And one of those farms, we had 1 colony of 0157 was isolated. And it was sent to the reference lab, and it was found to be exactly the same isolate. It had the same SNP profile as the second outbreak strain. And as you can see in this slide here, this is the watercress beds. The cattle were actually here, and this is a sloped field going down. So the sequencing data provided evidence of there being 2 separate outbreaks, both of which were linked to watercress. Strains from cluster 1 were not related to UK isolates, and this suggests a possible imported source, though we do not know the actual source of that. The other outbreak we showed, we resulted in finding an isolate from water, irrigation water, from one of the farms that grew the watercress, and therefore we think that contamination either came from runoff water from the cattle that were near that farm or from wild animals. So just in summary, then, whole genome sequencing is very good at identifying outbreaks, and particularly outbreaks that are either small in size and would not have triggered the exceedance, and those that are very geographically dispersed, or those that might have occurred over a very long time and would not have triggered our exceedance. They enable you to detect the outbreak more quickly because we're typing all our isolates and it's just a single method that we're using. And that means that there should be a faster time to resolution of the outbreak. It provides a more accurate outbreak definition. Peter mentioned this about refining the case definition in an outbreak. And whole genome sequencing is much more robust about saying whether one isolate is linked to another. And also it provides information on the phylogenetic relationships, so it can give you sort of point you or give you clues about possible sources or where that strain's come from. So it's making, having a huge impact on our outbreak investigations in the UK, and it's causing, they're also bringing about, more questions perhaps than we thought we knew how outbreaks worked and what was going on, but I think this is showing we clearly didn't, and we're getting a far better understanding of them. So just in summary, I'd like to acknowledge people who've contributed to this. I won't go through this slide because, as the others have said, there's so many people, but it's a huge effort across PHE to get us into using whole genome routinely for Salmonella. And just to say thank you for listening.